Good evening, everyone. It's great to see both new and familiar faces joining us tonight. Thank you for being with us, both virtually and in person. My name is Michael Wilson, and I'm the chair of the Invest in Research program at the Princess Margaret Cancer Foundation. I understand some of you were in a lab tour earlier this afternoon learning about DNA. I hope you found it of interest. I'm also sorry I can't be there in the foundation office. I've been working off a pretty rough cold and cough these last few days. So here we are. We've got a full agenda, so I'm just going to say a few words to begin. Before we get going on today's program, the Princess Margaret, the UHN, and the City of Toronto would like to acknowledge that we're on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis people. We often refer to the Invest in Research program as seed round funding for our researchers. Why do we call it this? Our Invest in Research supporters are helping our researchers take their first steps towards something new, similar to financially backing a new company. The first step is often the hardest. Our researchers have had no shortage of incredible ideas that have gone on to establish new research, new bedside practices, new technology, and so on. But they must be able to prove their research first, and the funds to do so are the hardest to come by. Over the years, we've granted out more than $3 million in Invest in Research grants. And while this sounds amazing, the true power of the program comes from what happens next. Our Invest in Research grant recipients have gone on to secure additional funds equaling close to $50 million more dollars. To put that into perspective, for each dollar that you invest in our researchers, they in turn have raised an additional $17. This sounds like a pretty good investment to me. Over the course of this meeting, you'll hear from last year's recipients as they give a final update on their respective projects. You'll hear from Dr. Aaron Schimmer, our Director of Research at the Princess Margaret, and you'll hear about 6 new and exciting projects that we can fund this year. I encourage you to ask questions at any time so that we can have an engaging conversation throughout the evening. Our goal is to fund the next big idea in cancer research. Every day, the Princess Margaret's renowned researchers bring creative and critical thinking to question the norms. They're using creativity to save lives and transform cancer outcomes around the world. And support from individuals like you make this happen. So let there be no doubt, investing in experimental cancer research is an unequivocal commitment to advancing medical science, pushing the boundaries of knowledge and fostering hope for groundbreaking treatments that can save lives and transform the landscape of cancer care. Thank you again for being with us tonight and for your critical support. Next, you're going to hear from Jared Drunowski. He's an associate director on the Major Gifts team and the lead for this program. Jared and I have had the pleasure of working together for several years on this project. He spent his entire career in the not-for-profit world with a focus on building and growing programs such as this. If you've not had the pleasure of meeting with Jared in person, I encourage you to do so. You'll definitely enjoy the conversation. Jared, over to you.
Thank you so much, Michael, and thank you to all of our Invest in Research donors who are with us both in person and online this evening. It's very easy to use phrases such as the world's best or cutting edge. But what does that really mean, and how do you back up those claims? Let me share with you a few examples of why we feel comfortable using these terms at the Princess Margaret. The Princess Margaret is ranked in the top 5 of the world's best cancer centers for percentages of published papers in high-impact journals. At any given time, we are running up to 850 active trials, and we have up to 23% of our participants on cancer trials. That is almost 3 times the national average. 96 patents have been issued to our clinicians and researchers, and we actually initiated the discovery of stem cells. These are just a few of the reasons why we are considered amongst the world's best. The Invest in Research program also supports some of the best and brightest minds that have ever worked at the Princess Margaret. Over the past 12 months, I've had the privilege of updating you on both current and former Invest in Research grant recipients that have had major publications announced, have won prestigious awards, and have secured additional funding for programs that were supported as many as 10 years ago. This helps prove that the work that you are supporting now will not only support clinicians and researchers today, but also for years to come. At this time, I'm also pleased to announce another amazing update regarding one of our Invest in Research grant recipients. Dr. Cheryl Arrowsmith, who will be giving us an update on her project in a few minutes, has recently been recognized for being amongst the top 1% of cited researchers globally. Let that sink in for a second. So let that sink in for a second. Of all cited cancer researchers in the world, our very own Dr. Arrowsmith is amongst the top 1%. This is an absolutely incredible achievement. Before we jump into our presentations for the evening, I'm going to take a few moments to run through some housekeeping items with you. Please note that we are recording today's session, and we will happily share this recording with each and every one of you post-event. To our guests on Zoom, we ask that you please mute your microphones and keep your cameras off during the presentations. However, we do encourage you to enter any questions that you may have on the right side of your screen in the chat function. We'll be monitoring that function throughout the course of the evening, and we'll be sure to bring up any of the questions you may have at the end of each presentation. For our guests in the room who have joined us today, we ask that at this time you mute your cell phones. And should you have any questions regarding any of the presentations, please hold them to the end of each presentation, at which time we will be happy to answer those questions. In the next day or two, our Invest in Research supporters will receive a link to a survey that will allow you to vote for the 3 recipients that you feel whose projects should be going forward and receiving funding for this year. We ask that you please complete these surveys by the end of next week, so that we can announce our 2024 grant recipients as soon as possible. At this time, I would like to introduce Dr. Aaron Schimmer, the Research Director at the Princess Margaret Cancer Center. In addition to his incredible leadership of the Research Institute, Dr. Schimmer has focused on developing new therapeutic strategies that target leukemia and leukemia stem cells. In 2018, Dr. Schimmer was actually an Invest in Research grant recipient. He and his team received funding that launched a study into the new ways to kill leukemia cells without harming normal cells or organs. They will soon be leading a first-in-human clinical trial to test a promising new leukemia treatment. His project has also secured an additional $2 million in support of his work. At this time, I ask you to please welcome Dr. Aaron Schimmer.
All right, it may seem a little less personal, kind of standing this far away, but let me just begin though by thanking all of you so much, whether you're here in person or virtually, for spending your evening with us here at Princess Margaret, and of course your contributions and support to Invest in Research and your support of research at the Princess Margaret Cancer Center. When we at the Princess Margaret invest in research, we make this investment into a number of areas. We support the scientists who are at the front lines making those breakthrough discoveries. We support the infrastructure to ensure our scientists are working with the most cutting-edge research technology. And importantly, and this is where this program comes in, we invest in early-stage research ideas. And I got to tell you, of all the areas, I'm particularly excited about those opportunities and particularly excited about the Invest in Research program. This is a way that we can invest in the earliest stage research projects, those crazy, maybe out-of-the-box ideas, but yet, if successful, have the opportunity to be paradigm changing in our diagnosis and treatment of cancer. Now, the investments that we make through Invest in Research are not, as Michael said, designed to fund the entire length or entirety of the project. Instead, it's really designed to get a leg up, move it to that next level, so it's going to be competitive for external funding. And now these days, with external funding being so difficult to come by, you need every leg and you need every advantage to put your project into that area. And to the testament of the success of the Invest in Research and to the testament of the success of our scientists, every dollar that we invest in research is leveraged 17-fold or more. And as Jared said, like I was fortunate to be one of the initial recipients of this program. And so I can tell you firsthand the impact it had. It was an investment made at a time when, frankly, the project was probably not at a point when external granting agencies would have been willing to invest in it. They would have said it's too early, it's too premature, you haven't taken the project far enough along. But thanks to Invest in Research, we were able to generate the data, move the project ahead so that others were able to invest in it. And then ultimately, it even attracted the interest of the NIH, the National Cancer Institute, who've now agreed to support a clinical trial based on this discovery, none of which would have happened if it wasn't for Invest in Research, because it would have laid in the back shelves of the research lab. And the story I tell you has been produced again year after year after year in other discoveries from our amazing scientists. So again, thank you so much for not just being here tonight, but your continual support of this program. It's critical to research at Princess Margaret. We've got three great scientists who are here with us this evening who are going to share with you their stories. These are last year's recipients, and so it's going to be kind of cool to see, and I don't even know what they're going to tell us, to see what they've done over the last year with this funding. And so the first I'm going to introduce Dr. Mohammed Mazhab Jafari. Dr. Mazhab Jafari is one of our senior scientists. His area of interest, similar to Dr. Arrowsmith, is in structural biology, meaning understanding the three-dimensional structure of proteins. And why that's important? That's critically important in the drug development and drug discovery process. You can think of drug discovery about trying to find the key that opens a lock. And I could give you a lock and I could give you 1 trillion keys, and I would say to you, which of these 1 trillion keys opens the lock? And you can imagine that would take you a very long time to test each one at a time. But what Mohammed's able to do is he can see the inner workings of the lock, so you can then manufacture a key that fits directly in it. And so Mohammed had a very exciting project that was funded last year. I'm delighted to call him up to give us an overview of some of the great accomplishments in the past year. Thank you.
Thank you, Aaron, and thank you everyone for your generous support and donation, which are really make a breakthrough for our projects, and it really helps us to gain additional funding, external funding, which are very, as Aaron mentioned, very competitive to get. So do I just go move forward? Yeah. So my group is interested in studying how cancer cells generate their own fat. These are specialized molecules called fatty acids. And the reason we would like to target these molecules is because in a healthy adult person, in a healthy adult body, our normal cells do not actually generate or synthesize, it's a chemical term, these fatty molecules, because we usually get them from our diet. So there is no need for our body to produce them. But in cancer cells, because they rapidly grow and divide, they need these specialized molecules to generate their cellular membranes rapidly. And if you inhibit this process in cancer cells, it effectively kills these rapidly dividing cells. And this happens in almost any solid tumors that you can think of. And this process is about 100 times more intensive in cancer cells compared to normal cells. So it's a great opportunity to target this chemical synthesis process. As many of you know, this is just a cartoon of tumor cells, if I can bring the laser pointer. This is a cartoon of a tumor cells. You can see this is a mass of a rapidly dividing cells. And these cells need these specialized molecules, which are called lipids. And they have a very interesting physical characteristic. So they're divided in really two sections. You can see the circle here is a region of the molecules that love water molecules. So we call it a water-loving region. And then the second region is a tail, and that's a water-hating region of the molecule. So because of this dichotomous characteristic, they can actually create different phases in the cell, so they can define the boundary of the cell compared to the bulk solution. And they organize into these massive aggregates, which we call biological membranes. So all of our cells are covered with these lipid biological membranes, and that's what they define them as a cell. And during the rapid growth of the cell, they continuously need to generate these fatty molecules. So in my lab, we use these powerful instruments, which are called electron microscopes, and they have a significant magnification capability. So they can magnify an image by 1,000,000-fold. So we can actually see individual atoms of the proteins. And this is a protein molecule that's involved in the fatty acid production. It's a very complex pathway, but this is the pathway that my lab really targets, how this takes these molecular building blocks and builds these fatty acid molecules. This is a general example of them. This is the water-loving head, this is the water-hating tail. So we're really interested to study this chemical process. And with the generous support from the foundation, we were actually able to solve the atomic resolution structure of the human fatty acid synthase protein complex. And we were able to solve it in complex with a very important anti-cancer drug called denifanstat, which is going on the clinical trial against multiple solid tumors. And now we're moving this methodology that we developed to study the structure of this important protein complex, to study how cells chemically modify it and how it is able to release its final product, which is a fatty acid molecule, into the cell. Basically, the question is that are there fatty acid acceptors in the cell that can accept the newly generated fatty acid? And if we can answer these questions, which we should be able to with the method that we have developed by your generous support, this could really give us more new avenues to target this important synthetic pathway in cancer cells. And with that, I would like to again thank you for your very critical and generous support that has enabled our studies. Thank you very much. So I guess now I will call. Oh, is there any questions? That's important.
So this is exciting work. Of course, it takes time to go from the discovery into the clinic, but everything worked out perfect. So if everything worked out perfectly, can you talk about the potential impact this would have on patients with cancer?
Yes, if this, let's say if we can actually discover an effective inhibitor, a small molecule inhibitor against the human fatty acid synthase protein, I think the immediate impact would be for patients with advanced cancer, because it's been shown that, at least in animal models with aggressive cancer, if you inhibit this synthetic process, it immediately results in tumor regression. Right now, what's inhibiting us to take this into actually a human case is the existing small molecule inhibitors have side effects because they result in weight loss, and which is unacceptable. So that's why we're trying to design a small molecule, or as you put a right example, a key for this protein that actually tunes down the activity of the protein and does not completely inhibit it. So if we find the right level of inhibition, I think that could make a huge impact, especially for the aggressive and late stage cancer. And this is in many solid tumors: brain tumor, colon cancer, lung cancer. So it could make a huge impact. Yeah. Okay, thank you.
So next, so next up, Dr. Naoto Hirano is our other recipient. Dr. Hirano is a senior scientist at the Princess Margaret Cancer Center, a cancer immunologist studying how to use immune cells, genetically modify them, and allow them then to recognize, attack, and destroy tumor cells within patients. Dr. Hirano's work has led to several high-impact publications and scientific discoveries. Importantly, though, his work has been licensed to pharmaceutical and biotech companies that are going to allow us to extend that into clinical trial for our patients. Dr. Hirano is currently in Hong Kong at a scientific meeting. He can't be here, but he sends his regrets, but he's also sent Dr. Lai, a scientific associate in his lab, who's kindly agreed to come and present his work and the work of the Hirano lab. And so, Dr. Lai.
Let me see the fancy slides. So thank you, everybody. And thanks to the Princess Margaret Cancer Foundation for funding this early stage work and the donors. So our work, and I'd like to also say Naoto sends his apologies that he couldn't make it today to present in person. But so essentially our work tried to address an unmet need in the treatment of a very aggressive cancer called acute myeloid leukemia, or AML. And so the treatment for AML actually hasn't changed in the last 40 years or so. And this is just showing survival curve data for those under the age of 60, actually. Survival actually has increased and has benefited from the use of hematopoietic stem cell transplants. And so survival over the decades have gotten a little bit better. Where for those over the age of 60, hematopoietic stem cells may not necessarily fit, or the specific recipients may not be fit enough to receive the transplants. And so transplantation is not working. You can see that survival is actually very, very poor. So immunotherapies have become sort of a mainstay, a very interesting treatment modality now for a number of different cancers, including blood cancers. And for the most part, I think all blood cancers now have some sort of immunotherapy involved in their treatment, except for AML. And so one type of immunotherapy is termed a bispecific T-cell engager. And so this is this molecule here. And essentially just adds, it's two different antibodies that are fused together. And essentially what it does is it tethers one side of it to a cancer cell, and the other side brings the immune cell towards that cancer cell. And so hopefully you activate the cancer cell and get some kind of killing. So BiTEs have been approved for the treatment of lymphoid cancers. However, for AML, they're currently in clinical trials right now. Unfortunately, the majority of BiTEs that are in clinical trial for the treatment of AML have also seemed to target healthy tissues. And so a lot of these trials have actually got suspended or terminated for the fact that the BiTE itself is actually harming the healthy tissues in the humans. And so we're getting a lot of unwanted side effects and toxicities. So the Hirano lab tries to solve some of these problems by targeting a different molecule, and one that's more, we hope to be more leukemic associated. And so this is a molecule called WT1. And actually WT1 is expressed on the inside of leukemia cells, but parts of the molecule get presented onto the surface of the cells in these little fragments in the context of this molecule called HLA. And so we hope that by developing antibodies and BiTEs against this particular target, we can minimize and increase the overall safety of the treatment. So with the funding we received, the first thing we did was just sort of ask the question of whether AML patients had actually detectable levels of this particular fragment of WT1 on the cell surface. And so in collaborations with Dr. Mark Minden and Andrea Arruda at the UHN Leukemia Tissue Bank, we were able to collect a few samples here and then actually show that the majority of the patients did actually show some expression of this molecule that we were interested in targeting. And so that was great. The detection and expression levels actually varied. And so you could have low expression, high expression, or middle expression. And so the detection actually was different, but we had something we could look at. And the other thing that we were able to do during this funding period was actually generate this compound in the lab. And so it took us a little bit of time to actually get the fusion together and the different construct together, but we were able to make a WT1 compound in the lab for sufficient activity. And we run these sort of leukemic killing assays regularly in the lab, where we take a leukemia cell and then culture it with a T cell and then throw the compound into the WT1 BiTE into co-cultures. And what we're just sort of measuring over time is just the killing of the cancer cell. And so we were happy to report that the BiTE that we generated in the lab was showing some activity. And so you can see that when we have a target cell that expresses the WT1 molecule, we did get specific killing in the presence of the BiTE. Except, and when you don't have the WT1 antigen present, we didn't see any activity. And so that was great. So one of the things that we are trying to do next then is essentially try to see whether this BiTE activity also affected AML patient samples. And that's sort of something we're trying to do immediately now. And so essentially to ask the question of whether if a patient has a low expression of WT1 on the surface, can we modify the BiTE in some way to get specific killing? And our immediate goal is to essentially collect enough data in the near future to be able to apply for external grant funding. And then this will help us generate enough preclinical data to further collaborate with industrial partners for hopefully clinical development. And so we hope that by targeting something like WT1, we provide a more safe and effective therapy. So the slides were sort of out of order. Yeah, I can take any questions and hopefully I can answer those. Yeah.
Hi, thank you. I have kind of a two-part question.
Yeah.
Number one is, are BiTEs used in immunotherapy for other types of cancer? And the second part is, could they be used to help make immunotherapy more effective for other types of cancer, like for solid tumors?
Yeah, so BiTEs are used for other types of cancers, and there's only one approved BiTE right now for the use of treatment of lymphomas against CD19 positive cells. So that's currently being used. And of course, like, I think the next step is essentially to try it in solid tumors as well. Yeah.
Other questions? Thank you.
Thanks.
So our next and final presenter is Dr. Cheryl Arrowsmith. Dr. Arrowsmith is a senior scientist at the Princess Margaret Cancer Center. She's the head of our protein structure and function group, and as you heard from Jared, is truly an internationally recognized and respected researcher. Dr. Arrowsmith, like Dr. Mazhab Jafari, is a structural biologist understanding the three-dimensional structures of proteins, but if I got this right, Cheryl, you're also a chemist in part by training. And so Cheryl also takes her knowledge of chemistry to structural biology and goes from that three-dimensional structure into drugs or chemical probes, one that can be used to understand the biology, but also leads for new generation of drugs. And I don't know if you're going to talk about open access science, Cheryl, maybe not. So one of the things that makes Cheryl's research program very unique is the open access philosophy. So all of what Cheryl discovers is made available to all researchers around the world for them to use and explore and work. The concept of working together, you can do more than one might do in an individual group or lab. So thank you, Dr. Arrowsmith.
Hi, great to be here tonight and give you an update on what we've been doing. So what I'll tell you about is an analogous to the talk that Dr. Lay just presented, where he's using antibodies that bridge an immune cell to target the cancer cell. And we're doing the same thing with drug-like chemicals, but that go within the cell and take a other cellular molecule, a protein, and bring it close to a aberrantly active, like a bad protein that's driving the cancer, and bring those two together so that the natural protein in the cell targets and degrades, causes the cancer-causing protein to be chewed up and put into the garbage. So this is, I'll go through this quickly. So this is what we set out to do. We're looking at the system, acute lymphoblastic leukemia, which especially in pediatric cases, when chemotherapy usually works initially, but a subset of the patients have recurrence. That recurrence very often has a mutation in this protein called NSD2. NSD2 is a protein we're targeting. And it makes, one of the reasons it's so bad is it causes the cancer cells to go to the brain, and it can't be treated then. So that's what we're looking for. And it also causes the cells to be resistant to the chemotherapeutic drug dexamethasone. So we're going to try to stop that process by stopping the leukemia cells before they get to that stage and killing them. And so we're doing it, as I mentioned, with these chemical tools that constitute a new exciting drug modality called targeted protein degradation. And the way we do it is by putting tagging the protein of interest that we want to get rid of, so that this other protein in the cell will modify it and send it to the cellular trash bin, if you will. So what we do is we'll be measuring leukemic cells, and in the presence of these molecules that we developed a series of molecules with our chemistry collaborators to test whether they can achieve this. Whoops, sorry. And the long-term goals would be eventually see if to provide the proof of concept that this type of strategy could be taken forward into a therapeutic modality and provide a better quality of life for cancer patients, particularly children where this happens. So with funding from Invest in Research, thank you very much. We published this paper earlier this year that identified a new way to tag the aberrant NSD2 cancer-promoting protein. And at the time when we published this, the study was in the works, and then your funding helped us sort of push it over the line and be able to publish this paper. We still didn't really understand what the mystery protein was that was causing the degradation and shipping the protein off to the garbage. And over the last 6 to 9 months, we've been able to figure out what that protein is, the garbage truck protein. It's called FBXO22. So we developed improved the tag that enabled us to identify FBXO22, which targets NSD2 to the garbage. And at the same time, we showed that this newest tag degrades NSD2 and kills those cancer cells that have this mutant NSD2 in it. And it also, in so doing, reinstates or overcomes the resistance to dexamethasone. So this is exciting for 2 reasons. It helps validate NSD2 as a mutant NSD2 as a target in this disease. And so this is a personalized medicine or targeted therapy, if you will. So we're a very specific type of cancer can be targeted with a drug. And so we think this validates a further investment in this strategy. And it also has wider implications for the whole field of targeted protein degradation, where one wants to target, there's many mutant proteins in cancer cells and in different mutations and different types of cancers. And now we have a new tool to try to target them as well. So that's, we're very excited about that, and we appreciate the support of Invest in Research. Okay. And research is a team sport, and this is the team, my great team that you helped support. So 2 PhD students, a postdoctoral fellow, and my wonderful lab manager, Sheely Dwan. We work a lot with my colleague here at Princess Margaret, Brian Rout, who helped us find using proteomics studies to find the FBXO22 protein. My chemistry collaborators at the University of North Carolina, and Dr. Jonathan Licht, who runs the University of Florida Cancer Center, and he had the models for the ALL with the mutant NSD2 in it. Thank you. Happy to answer questions.
Dr. Smith, quick question for you. It seems like you've made a lot of progress in this field. What do next steps look like for you and your lab with respect to this project?
Yeah, we want to, so we have a grant application pending to CHR to further fund, and I think we're very competitive for that grant now, to try to, there's so many questions to answer, like, so if we, does our small molecule, does it interfere with the normal activity of FBXO22 so we don't want to disrupt the cell in some other way. But mainly further developing the small molecule to be a safe and effective targeter of, in the first instance, ALL with a mutant protein, but there's this protein is mutated in other cancers too, particularly lung cancer is one where we think we would like to look at. And we have great colleagues within Princess Margaret who have all these great lung cancer models, Ming Tsao and his team. So there's a lot of work we can do in additional cancers, and then also understand how to make new target other mutant proteins as well with this strategy.
That's right, exactly. All right, so first of all, thank you so much, Mohammed, Dalam, and Cheryl. Thank you so much for taking your time to present with us, and amazing what you've been able to do in a relatively short period of time. All right, I got you. So I have the privilege of being able to speak at a number of events on behalf of our scientists and with the foundation. But I have to tell you, it is this event that causes me the greatest stress and where I feel the most pressure. And the reason I feel such pressure is that the scientists have, put their faith in me to try to present their work in the best possible light. So I'm going to do my very best to convey the excitement and enthusiasm for these 6 projects. This year, and similar to previous years, we had over 30 applications for the Invest in Research program. It interestingly comes from across the organization, from newer scientists, from established individuals, from people who are PhD level scientists, who are clinicians, and even, as you'll see this year, from people outside maybe our usual funding streams, a psychologist and a medical physicist. These 30 applications were reviewed by a panel of our scientists. They evaluated the merits of the proposal and selected the top 6 that they felt were the most scientifically meritorious. Those 6 are now coming forward to you for your decision. I'll tell you and share with you what I thought were my top 3, but that's only my personal opinion. What I would say to you is you cannot make a wrong choice. They're all excellent, they're all good, and so whatever speaks to you for whatever reason, if it's the disease site, if it's the science, if it's the commercialization or the therapeutic potential, whatever you use to choose by, it's all good. You can't go wrong. All right, so if you like the presentation, I'll take credit for it. If you don't like it, don't penalize the scientists. Look beyond my presentation. All right, so the first proposal is by Dr. Stephen Chen. Dr. Chen is a senior scientist at Princess Margaret. He's also a clinician, and like me, he's a member of the leukemia site and treats patients with acute leukemia and related disorders. And Dr. Chen is trying to develop a new treatment strategy for a group of patients with very high risk acute leukemia who do not respond well to standard treatment options. And what Dr. Chen has discovered is that these particular type of leukemic cells have an antenna that sits on the surface of the leukemia cell. The antenna is called CD59. And what he's discovered is that CD59, that's the name for this antenna, is important for the growth of the leukemic cells. And it allows the leukemic cell to resist chemotherapy when given to the patient. In addition, Dr. Chen has shown that when you genetically deplete or knock out or take out this antenna, the leukemic cell dies. And so this becomes a very interesting project. I think this is, again, one of my top 3, where Dr. Chen has proposed to understand if and how this antenna is important for the growth and survival of leukemic cells, both in experiments done in Petri dishes and also to be done in mouse models. The second part of the proposal is going to look at strategies to knock out or get rid of this antenna that sits on the surface of the leukemic cell. And what's particularly exciting about this project is it's known that there is a bacterial enzyme complex that for other reasons is capable of degrading or eliminating CD59 in other circumstances. So what Dr. Chen wants to do is test whether this enzyme complex, in the context of leukemia, in petri dishes and in mouse models, can degrade this complex and kill selectively leukemia cells. This work is important for a number of reasons. It's going to uncover new biology about why leukemic cells grow and why some are resistant to chemotherapy. And it's important because it'll address or identify a new treatment strategy for a subgroup of patients with very aggressive disease. And it's important because the route that he's discovered for this potential therapy has the ability to advance rapidly into clinical trial because some of these therapeutics are already used for other indications. So again, a project by Dr. Chen, a clinician scientist, looking to understand the importance of this antenna CD59 on leukemia cells. Questions? Oh, yes, go ahead.
Is CD59 found in any other cancer tumors or cancer cells?
You know, interestingly, Dr. Chen has shown that there are some other subtypes of cancer, so some groups of breast cancer and some other solid tumors that also have this. So, you know, what's interesting is although his models are in leukemia, because like me, he's a leukemia clinician scientist, this work will have relevance for cancers beyond leukemia. Again, sort of often highlighting what we see in science, that learnings in one cancer type can sometimes have applicability in others. So that's a good question.
What's the current standard treatment for this type of AML?
So the standard treatment, if you'd open the textbook, would be essentially a 2-drug regimen that I would argue is largely unchanged over the last 35 years. The problem is that this 2-drug regimen will produce a remission, will be able to get rid of the leukemia in about, depending on the patient, 25 to 30% of cases, so meaning 70% don't respond. And in virtually 100% of those cases, the leukemia will come back if treated with only of chemotherapy. So we are working, as you can imagine, on developing new standard chemotherapy approaches, new novel treatment options. And while many of these are used or show promise, patients continue to relapse even after this treatment. So there's a large need for an effective therapy for this very high-risk group of patients. All right, good. So we will move on. So I don't know if they're putting them in order or not, but this is another one of my top rated. This is number 2, actually, of my top rated applications. I'll tell you the next one, the top one, a little bit later. This is work by Dr. He. Hansen is a senior scientist at the Princess Margaret. His area of interest is in what we call RNA medicine. So being able to deliver genetic material to cancer cells to essentially correct, edit out the defects, the genetic defects that are inherent of cancer cells. And by doing so, either flip those cancer cells into normal cells or eliminate the malignant cell. And this represents a new generation of therapeutics for cancer. Its origins are actually in the COVID vaccines. So the COVID vaccines, as you may know, because we all became scientists during the pandemic, if you recall, these are a nanoparticle, an mRNA nanoparticle, an mRNA vaccine. So what's an mRNA nanoparticle? It's a ball of fat, that's the nanoparticle. And then you've got the mRNA, and when you inject the COVID vaccine into your arm, the mRNA makes something called a spike protein, and that is a part of the COVID vaccine. It's not the, that's part of the COVID virus. It's of course not the whole virus, just one tiny part of it. In and of itself, that's not infectious. And the body then recognizes that foreign protein and makes antibodies, and that's what protects you from getting COVID. Now, it turns out these nanoparticles were actually originally being developed for the treatment of cancer. And this is why when COVID happened, we were able to develop vaccines within essentially 1 year of this disease becoming apparent, because they were so far advanced for cancer. Now, these vaccines that we currently use for cancer, they have challenges because the nanoparticle preferentially goes to the liver and doesn't really go to the cancer. So there are two strategies that you can employ to try to selectively deliver a COVID vaccine-like nanoparticle and genetic material to a cancer cell. First of all, you can change the nanoparticle so it better goes to the cancer cell, or you can change the genetic material in the nanoparticle so that it is only produced inside a cancer cell. And what Dr. He is doing is taking a two-pronged approach to target lung cancer. So he, with his collaborators, have developed a nanoparticle that you can inhale and preferentially goes into the lung cells and the lung cancer cells. And they've also added a component to the genetic material in this nanoparticle so it only gets essentially turned on in a lung cancer cell. And so this is very novel in terms of an approach. It has the opportunity, of course, to be a game changer in the way that you treat lung cancer. And the same kind of technology, you can also think, we had a question, is it applicable to other cancers? This can be applicable to many cancers. What you would need to do is fine-tune the mRNA genetic material so it only gets turned on in the cancer cell you want. This is an idea, and it's an area where Dr. He has experimental and scientific expertise, but yet he's not had the point to be able to move this to the point of demonstrating the success. Potentially through funding through a program like this, if he can generate that preliminary data, there's no question in my mind it will be set up for being a highly competitive external grant and have the potential for commercial investment. And this is an area you could move rapidly into clinical trial, similar to Dr. Chen's discovery.
Aaron, it's Michael. Could this not be perceived as a potential replacement for chemotherapy treatments down the road?
Yeah, you know, this is what's so exciting about this whole area, that if you could selectively eradicate cancer cells without the use of chemotherapy, you can bypass most, if not all, of the side effects that we traditionally associate with chemotherapy, the hair loss, the infections, etc. So this is the hope. I mean, obviously that's a bit of a longer term goal, but this is what this technology can offer. It's an area we're making huge investments in at the Princess Margaret, and Dr. He happens to be leading that whole area.
Thank you.
Dr. Shimmer, quick question for you. How are these RNA drugs being produced so quickly when it seems like many other drugs take years and years and years to produce?
You know, so that's the excitement around this technology. So it's leveraging what was happening before COVID, it's leveraging the lessons learned in COVID in the ability, I mean, think about how many billions of doses of nanoparticles that have been produced in the world up until now. So we are really good in this world at making nanoparticles. It's relatively straightforward to make changes to the nanoparticle that can direct it elsewhere. And you can swap in or out different genetic material just like you do for the COVID vaccine. And because of what we've learned through the COVID vaccine allows us to move very rapidly on this technology, in some ways a little bit different than what a small molecule drug, which, you know, they're very important to do, but they can take 10 years to go from idea to an approved drug. Okay, so the next presentation is from Dr. Jelena Lukovic. Dr. Lukovic is one of our new clinician investigators in the Department of Radiation Oncology and is proposing to study a new form of radiation treatment for patients with larger and bulky tumors. And in many patients with cancer, particularly those with advanced cancer, at some point in their cancer journey will receive radiation therapy. In some cases, it's used as a curative modality, but in fact, in the majority of cases, radiation is given as part of supportive care to relieve symptoms from larger, bulky tumors that are pressing on structures within the body. If you can shrink that tumor, of course, you can alleviate many symptoms that a patient is experiencing. One of the challenges, though, when dealing with a large, bulky tumor is how to give enough radiation to kill the cancer cells while at the same time not providing radiation to the surrounding tissue. That's a real challenge. And what has been shown is that if you adjust the way the radiation is delivered, if you adjust that beam of radiation, you can deliver high doses of radiation to the center of the tumor, lower doses to the periphery, which are sitting nearer to normal structures, and produce still significant shrinkage of the tumor. This is a technique, it's called spatially fractionated radiation therapy, or SFRT, but of course that's what you would call it, right? And this is this technique. And it's a technique that's been evaluated in other centers in early phase clinical trial and is showing very promising results internationally. But there isn't a program in Ontario that's delivering this modality for our patients, either here at Princess Margaret or across the province. So this proposal is designed to establish this modality here at the Princess Margaret and do it in the context of a clinical trial where they will evaluate the effectiveness and side effects of giving this treatment. It's an important treatment modality. It could have significant impacts for patients, especially those with larger tumors that are sitting near vital organ structures. And again, Dr. Lukovic is an expert in radiation oncology and will have the skill sets to advance this type of treatment option. Yeah.
Do the machines that deliver radiation right now have the capability to deliver it in this fashion?
Yeah, very good question. Yes. So this would use existing radiation linear accelerators, the equipment that's currently available at the Princess Margaret. It's also available at other cancer centers. It's not unique to us. But it's actually how that radiation is then delivered using the existing machines that makes this approach different than standard radiation, which basically would cover the site with the same dose.
Dr. Schimmer, you mentioned that we are not currently doing this type of treatment at the Princess Margaret. Is this being done elsewhere in the world that you know of, and do we know if it's safe enough to do here as well?
So it is being done elsewhere in the world. They have evaluated it in early phase clinical trials where it is showing promising results. So this is a mechanism by which we could begin to develop this modality here at Princess Margaret for our patients. So this is, the next proposal is by Dr. Tracy McGaha. Dr. McGaha is a senior scientist, a cancer immunologist at the Princess Margaret. And you know, what's interesting is you've just heard a very clinically oriented project. This is going to be one much more around fundamental discovery. And this project is using spatially defined analysis of TCR and BCR repertoires for neoantigen discovery in pancreatic cancer. And Jared, I'm not sure I need to even say more about that project. That's just so self-explanatory, right? So anyway, okay, so pancreatic cancer. So many of you may know pancreatic cancer is probably one of the most aggressive and difficult to treat types of cancer. Despite many efforts and different types of chemotherapy, response rates or ability to shrink the pancreatic cancer cells with chemotherapy is low. Even when successful, it rapidly regrows. And although pancreatic cancer can be curable if picked up very early, unfortunately, in the overwhelming majority of cases, pancreatic cancer presents much more advanced and much later in the course. And so, you know, acute leukemia is one of those diseases, but certainly pancreatic cancer is too, a disease with a very large unmet need. There are some emerging signals that immune therapy may be successful in this disease, and there are some emerging clinical trials, particularly, that are using cancer vaccines to target pancreatic cancer. There's been one very recent study that's shown preliminary success. And what Dr. Magaha wants to do is a new way to identify the targets for future cancer vaccines or future cell therapy against pancreatic cancer. We talked a little bit a while ago about the COVID vaccine. And what you may know or may have heard of is that the vaccine is directed against part of the COVID virus called the spike protein. And it's the ability to identify the spike protein that allowed us to ultimately go on and make the vaccine. Because you had to identify the most critical part of the virus. If you made the vaccine against other parts of the virus, it probably wouldn't have been successful. So what Dr. Magaha is doing is he's looking at the pancreatic cancer cells and trying to understand what are the unique proteins that distinguish pancreatic cancer cells from normal cells. Because of course, if you make the vaccine and the immune system recognizes all normal cells in the body, you've got a major problem. So you've got to find proteins that are only in the pancreatic cancer cell. And he's doing it by looking at the immune cells and what they are seeing, but not effectively being able to target. So it's a new way to approach cancer vaccines for pancreatic cancer. It's more at a fundamental discovery stage, but the advances here could be paradigm changing for future treatments and are going to tell us a lot about the biology of pancreatic cancer and why even immune therapy, although early successes, is why doesn't the body's own immune system recognize this disease? So lots of exciting things that are happening, understanding the biology of a very aggressive tumor.
Dr. Sherman, this is very exciting to me. It's Michael again. You know, I know of two people, long term, that have survived pancreatic cancer, that had surgeries. In an experiment like this, is research done on people like that to understand their molecular structure as to why, and would that be helpful in investigating those kinds of survival patients?
Yeah, that's a fantastic question. You know, there's a whole line of research that's looking what we call exceptional responders. So people whose cancer has done much better than would be expected, to try to understand what's unique about that. I must say that this project per se isn't addressing that particular question, although it's a good question, but would hopefully perhaps turn more of patients into those exceptional responders if this type of therapy works well.
Thank you.
So this is a proposal from Dr. Rinat Nissim. I was talking to you about how we've received applications across the spectrum of the Institute. And Dr. Nissim is a psychologist. And so it's great to see like how this program is resonating. As an aside, you know, our scientists, our clinicians, they're busy people, and they're not going to bother putting applications in for things that they just don't think are important or worthwhile. The fact that you get 30 applications across the entire Institute, you know, there's a signal that this is an important program. And Dr. Nissim's work focuses on caregivers. So this is a hugely important and probably understudied aspect of the cancer experience and cancer journey. As I'm sure you know, as I know, you know, that the patient undergoing treatment, undergoing the cancer journey, I mean, obviously it's a very difficult time, but you know, so is it for the family and their support system, the caregivers who are at home with the patient. You know, they have significant burdens, whether it is the emotional burden, the physical burden of helping care for individuals, and the financial burden. So this kind of thing is tough. And work by Dr. Nissim is actually led, and here at Princess Margaret, we actually have the first of its kind in Canada, which is a caregiver clinic. And it helps address the needs, the psychosocial needs and provide support for these caregivers who are so crucial in a patient's journey. And what Dr. Nissim is proposing is to really understand more about the challenges that caregivers face. You know, I gotta tell you, we spend a lot of time asking patients about their experience. I'm not sure we ask caregivers as much. And so what Dr. Nissim is going to be doing is conducting focus groups of caregivers, and particularly caregivers for whom English is not their first language. Because when you think about the burden of patients whose language is not the first language, and caregivers who's not the first language, it's tough, but they've got even a tougher experience. So she's going to be particularly focused on the caregivers who don't speak English as their first language, understanding what are the challenges that these individuals face. Because by understanding what their experiences are, it's possible then to develop new interventions to help this important group of people.
Dr. Schindler, it seems to me to be fairly obvious that we should be asking these family caregivers, but we haven't been, unless I'm wrong. Is there an example of a time or a scenario where we have been asking family caregivers and we've come up with a better way of dealing with this group of individuals based on our feedback?
Yeah, you know, look, I'd be the first to acknowledge that we probably do not spend enough time understanding the impact of cancer on the caregiver. I'll tell you, in my clinic, I think we spend most of the time asking about the patient's symptoms, how's the patient doing? I could probably spend more time asking about how the caregiver is managing. But it was through studies like this, it was through initiatives like this, that the caregiver clinic was established. And I think what's particularly good about it is in a busy oncology clinic where we really are probably focusing a lot on the patient and their often physical symptoms, there's a place that the caregivers can now receive the support that they need. And that came out of focus group studies early on with caregivers. This has an opportunity to extend that, provide additional services that we're going to provide caregivers in particular, as I said, the group that may be particularly challenged because of language barriers. All right, and so we'll end with a proposal by Dr. Thomas Purdy. This is actually my top ranked application, I'll tell you. Again, all of them are good, you vote your conscience, but this was my top ranked proposal. And Dr. Purdy is a medical physicist. So medical physicists are those that plan the radiation therapy that patients receive. They operate the radiation machines. Very specialized group of individuals. But Dr. Purdy is also an international leading authority in artificial intelligence and machine learning. In one of his recent, well, let me take a step back. When patients have radiation planning, they undergo a series of CAT scans, and then the medical physicist with the radiation oncologist carefully looks at all of the CAT scans and defines exactly where the radiation beam should go and not go, so you hit the tumor but not the surrounding normal cells. What Dr. Purdy showed is that you can use machine learning and artificial intelligence to do what a radiation oncologist and physicist would spend hours doing. It can do it in a few minutes. And it can do it as well, if not better, than the trained professionals. So this work has had major implications. It's got commercialization potential. It can save the system money. It can allow us to see more patients because you can more rapidly do things using artificial intelligence. Now, Dr. Purdy then wants to take his expertise in artificial intelligence to address another problem in the radiation planning process. So I must tell you, I'm not a radiation oncologist, so I actually didn't even know this until I read the proposal. But it turns out when patients are undergoing radiation oncology, radiation treatment, you can't use the CAT scans that were done at the time of diagnosis to plan the radiation. And you can't, because the radiation, it depends on how you hold your hand, how you breathe in. You got to direct the beam in such a way. And if when they were doing the CAT scans for diagnosis, your hands were at the side and not over your head, it can change a little bit the way the tumor is positioned. And if you haven't accounted for that and you deliver the radiation, you could start to hit normal tissues. And so what happens is the patients have to undergo a whole new series of CAT scans, but with their hands held in different positions, breathing in, breathing out, all to map exactly the way the tumor would look when they deliver the actual radiation. So not only then do patients have to come back for more radiation, more CAT scans, it can take several weeks from the time you've had your initial scans to you have these second scans, and that costs the system money. It takes time. It's the emotional distress that you can't start your treatment right away because you got to go through all these other scans. So what Dr. Purdy has been able to do, he takes his expertise in artificial intelligence, machine learning, and can now use those initial diagnostic scans to be able to model what it would look like if the arm was this way, the arm was that way. And so patients now can have their radiation treatment based on those initial scans. And so Dr. Purdy has got the expertise to do it. He's presented some data to demonstrate that his approach would be feasible. But this grant, this application is designed to get it to the next step so that external agencies would realize that this is really a feasible opportunity. If this is successful, this would dramatically change the way radiation therapy is delivered. It would save our healthcare system significant amounts of money. It would allow patients to have faster times to treatment, and there's a huge commercialization potential for this technology. He's done it once before in this field, and he's poised to do it again. It's a great proposal.
Dr. Schimmer, if I heard you right, does this work deal with the exact patient's earlier scans, or is it artificial intelligence based on that patient plus a subset of people that would look like him, like hundreds of others, or is it really tailored to the exact patient?
So this is designed so that when an individual patient has their scans done, on that patient, you can do the radiation planning without the need for second scans. So what are they going to do? They're going to take tons and tons of original scans. They're going to take tons and tons of scans that were done like with the arm this way, the arm that way, and use them to figure out how from the initial scan, what adjustments you have to make for the arms being held in different ways. It seems so simple at some level, but it's actually really not. You've got to use complex mathematics and machine learning to be able to make those adjustments. But I think there's good reason to believe that that can be done.
Thank you.
Dr. Schimmer, Dr. Purdy's one-page synopsis focused on prostate cancer, but given that it's machine learning, is this technology, is this method limited to prostate cancer, or could it be transferred to other cancers as well?
Yeah, no, not at all. It's actually widely applicable. He's given you an example, just based on feasibility. You can't do like literally all cancers, but this should be able to, you'd have to go through the step with each and every cancer, because I'm sure there's subtleties between a breast cancer and where it's located and a prostate cancer. But the concept is that this could be widely applicable. And just think of all the patients with cancer who need repeat CAT scans, the time that takes for the patients, the emotional burden and the cost. Like, this could be a game changer.
I'll be really loud and short. As you mentioned, it's fewer CAT scans on our health system, so that actually opens up opportunities for treatment for other patients with diagnostic tests. Sometimes there can be wait times.
Oh, yeah.
What are the patients using foods that are needs a scan, and how do you prioritize that so that the savings and the benefit is really interesting.
Yeah, absolutely. There's a whole trickle down effect from this.
So this is maybe a bit saucy of me, but with the incredible benefits this could have to our entire health system, it makes you wonder, why doesn't the government fund this kind of work? Because they don't, right? It comes down to you and the Tom Purdys of the world.
So there are different answers. So the answer most relevant for the Invest in Research panel is, this is a great idea. Tom Purdy is an expert in the field. He's provided the rationale for why this would be feasible, but he's not yet competitive for external funding or government funding, because he hasn't quite gone that next step to really demonstrate this in at least a small group of patients, right? This is what this funding will do. The other answer to this is also disappointing, or maybe it's appropriate, I don't know. Healthcare dollars, not a single dollar of healthcare dollar can be used to fund research. And so whether it is in fact the lighting or the heating that goes into the research floors, even in the hospital, not a penny of that expense can be attributed to healthcare dollars. There's, and this is, I understand why, there's a real firewall, and so this is why the government will fund it. This is too early for them. It's got to be proven as effective therapy.
noticed one of the earlier presenters, had like a collaborative effort. Like, are these all here, or is it collaborative, some of them, or not that it matters, as long as we get the answers, we're happy, but just wondering.
Well, all of them are collaborative. So no one does science alone anymore, at least not good science. But in all fairness, and this may be a deciding factor for you, some are going to be more collaborative than others, meaning that some involve other labs at the Princess Margaret. Some are going to pull in international expertise with the needed. Others will be collaborating with others in Toronto. So yes, I mean, there are some that are going to be more collaborative in that regard than others, but none of these are silo efforts. All right, so I've tried my best to give you excitement of all of them. Again, if you didn't like it, make sure it's not because of the way I presented. I don't think you can go wrong. They're all great projects. Whatever it was that spoke to you about the one that you like, that's good. And in the end, you should vote your conscience.
So ladies and gentlemen, you've now heard about 6 incredible proposals, any one of which is deserving of funding, as Dr. Schimmer said. Unfortunately, we can only choose 3 of them for the coming year. So in the next few days, you will receive an email that has a number of attachments to it. We will send back out the one-pagers, the synopsis for each of the projects. We will also send out a survey link, which will allow you to go in and vote online. We will also be sending out a recording of today's presentation. So if at any point you want to go back, listen to a specific presentation, read the one-page synopsis, please feel free to do so. We will ask that all voting take place by end of day next Friday. For obvious reasons, we'd love to let our finalists know who is being selected for the 2024 Invest in Research grants. At this time, I would very much like to thank Dr. Schimmer and his team. Susanna Moore, Patrick Yao are with us this evening as well. This is an incredible team, and this evening simply could not have happened without them. Please, a round of applause for Dr. Schimmer and his team. I also want to thank Michael Wilson, who is online with us today. Michael has been an incredible chair for this program, an incredible partner for myself. Michael, your dedication, your enthusiasm is unmatched and unrivaled. I can't thank you enough for your drive in this program and for continuing to push all of us forward with Invest in Research. Lastly, I'd like to thank all of you. The Invest in Research program, as you've heard, is one of a kind. The individuals that we have supported over the past many years have come through with incredible research and have really pushed the envelope in what Princess Margaret can do. None of this would be possible without you, the supporters. So thank you again. Thank you for joining us this evening, and we hope you have a great rest of the night. Take care.